sequences encoded. And they will add themselves at what is called plus end. It'll grow from the plus end. Okay, once we get the tubule started, then new units are added to it from the plus end and it will grow. And the other end is a minus end. Okay, now this is usually, these microtubules are usually located, the minus ends are close to the nucleus, and they radiate out toward the cell periphery, okay, in general, all right? As I said, they're hollow on the inside. These are the biggest fibers. Then there's something called microfilaments. These are the smallest of the three groups of fibers. And these, once again, are made up of a special type of protein, a globular protein that forms a solid rod. And this protein is called actin, okay? Actin, all right? And there's several types of actin, but we're not going to differentiate between them. But two fibers of actin, two filaments, will come together and they're sort of wrapped around each other in a coil, right? Likewise, these are polarized, plus, minus ends. These tend to go around the cell, inside, okay? And I'll talk about their functions here in a moment. Okay, so these are, of course, huge structures made, they're polymers of, okay, the microtubules are polymers of alpha and beta tubulin. I mentioned when I first introduced polymers, it was at the macromolecule level, but we have higher levels of polymerization, okay? So there's thousands of these proteins that are in the microtubules, okay, typically. And likewise with the microfilaments. And the third type are called intermediate fibers. This is sort of variable as to the proteins that compose them, and they tend to be the more permanent fibers in the cytosol. A lot of times they're made up of keratin. That's also a structural protein, but there's other proteins that may be involved as well. And they tend to give the cells shape, they're more permanent. Whereas the microtubules and the microfilaments, they are rather dynamic because they form, disassemble, and form, and disassemble, okay? All right, so let's go back 2 slides, and what are their functions, okay? So this is an electron micrograph showing this cytoskeletal material, okay? So this is an electron microscope. So some of these proteins are framework, scaffolding for the cell, gives the cells their shape. And some of it is support, okay, for various things. But what is real important here too is movement. Those transport vesicles that are moving from the rough ER to the Golgi to the plasma membrane from the rough ER, that is not random motion. They are walking along trackways. Those trackways are formed by microtubules and microfilaments. So for those transport vesicles to move, however, there has to be a motor protein. And so there's also motor proteins here, 3 types. All of these have subtypes, okay? Kinesin, dynein, myosin. Myosin is actually a large family, okay? There's about 11 different classes of myosin, each of which have numerous, what are called isoforms. And so we have a lot of genes for different types of myosin. Skeletal muscle uses a different type of myosin than what you typically find as a motor protein here, okay? So these motor proteins are really interesting, okay? Because all 3 of them, at least for moving traffic within the cytosol of the cell, they have similar structures, although they're made up of totally different proteins. So they have leg domains, 2 of them, okay? And a domain that holds the legs together and a cargo attachment domain, all right? And they can walk on microtubules. They can walk on microfilament trackways. The motor proteins that walk on microtubules, kinesins and dyneins. And so, and it's the myosin motor proteins that are walking on microfilaments, right? And they'll have a picture, I'll show a picture here in a moment. But if you have here a microtubule, a transport vesicle, stuff inside. Then here we have cargo, load domain, leg domain, another leg domain, and these can alter shape. Now, it's not random, it carves in ATP. So when ATP is used to phosphorylate this structure, it'll cause one leg to move forward, and then when the phosphate is removed, another conformational change, and so forth. ATP after ATP, these things are walking along the microtubules. Okay? Now, as I said, show a picture here, just a second. Yeah, and so this is sort of cartoon up here that's showing the motion of kinesin. This is a microtubule, and these legs are moving in a certain direction, directed direction. Inside of the cell, I'll draw it over here. Inside of the cell, I have the nucleus here. The microtubules tend to be coming from one spot close to the nucleus, which I'll give a name to it momentarily, and they're heading out toward the periphery. The kinesins move cargo from the cell center to the periphery. They're always moving in that direction. The dynein motor proteins are going just the opposite on microtubules. Okay, dynein is moving endosomes, okay, that have butted off of the plasma membrane, bringing stuff in. They would be moved toward the cell interior by dynein proteins. As far as the actin and myosin motor proteins, these tend to be trackways that go through the cell and the myosin proteins are walking around. Okay. Later, when we get to lab exercise 6, you'll be looking at plant cells and you'll be able to see chloroplasts. And if you get the right type of preparation, those chloroplasts are going to be moving around the cell periphery because they are being moved, they're walking, the chloroplasts are cargo being transported around the cell on microfilament trackways by myosin proteins. And you'll get to be able to see this motion. Okay? All right? Cytoskeleton. 3 types of fibers. There's other structures that are found typically in animal cells too. Cilia and flagella. Okay, the flagella are, the main differentiation here between cilia and flagella are their length. Flagella is long and cilia happen to be short. However, there's more to it than that. Okay, there's other differences here, but I'm not going to get into it. As far as their structure, however, okay, well, first of all, what do they function as? Well, obviously movement. Certain unicellular organisms, such as paramecia, you'll get to see some live paramecia as well, covered with cilia, and by the beating of the cilia, this is how they move, okay? Furthermore, sperm cells in animals have a long flagella, and this is how they move, by the movement of the flagellum. But it's not only just on the movement of cells, it's also stationary cells to move fluid across the cell surface. Okay, so for example, in your respiratory tract, the lumen is covered with cells that have cilia, and they're constantly moving to move mucus that are secreted up the respiratory tract into your throat. Also, if you have a stationary protozoan, it's heterotrophic. It has to be taking in little particles of bacteria in the pond that it happens to be in. If it can't move, it has to refresh the water around it because eventually it will exhaust everything that's there. And so the beating of the cilia will move that along, even though the cell stays in the same part. So that's something that the cilia happen to do. Now, as far as their structure, I mentioned already, flagella in bacteria. We're using the same word for something that's analogous, but not homologous. Okay, these have totally different structures and totally different evolutionary origins. As I mentioned with the bacteria flagella, it is a solid rod of protein made up of flagellum, multiple units of flagellum. This here is different. In eukaryotes, the ultrastructure of a flagellum and cilia happen to be made of microtubules. Okay, so if we took a cross section, let's go ahead and take a cross section. If I have, well, okay, I'll come here. I'll go back in a moment. If we took a cross section through a flagellum or a cilium, we would find that there are 9 doublets of microtubules around the periphery. So what do I mean by a doublet? We have 2 microtubules that share a common wall. Okay, 2 microtubules that share a common wall. And there's 2 separate microtubules right in the middle. So these are running the length of the flagellum or the cilium. There's other proteins in here too that hold them together. Okay, so there's spike proteins. We're not going to worry about them, but these are microtubules. Just like the microtubules I talked about that are trapped ways inside of the cytoplasm. Furthermore, the doublets are attached to each other by dynine motor proteins. Okay, so we have a cargo-bearing regime and also legs, but in this case, the cargo domain is firmly attached to one of the microtubule doublets, and when ATP is used, this will walk along the adjacent microtubule. And this is how they move. Now, there's a problem here. If we have 2 microtubules, these are dynine, this here is attached to this microtubule, and it's going to walk up like that. All we would end up doing is moving microtubules up and down. Okay, that would not cause any sideways motion of the flagellum or the cilium. However, at the base of the flagellum and cilium, these microtubules rearrange themselves into something that's called a basal body. And I'll show a basal body. Oh, this is a basal body down here. We have 9 triplets, okay? Nothing in the middle, but 9 microtubule triplets. When I say triplet, there's 3 microtubules that are sharing common walls. Furthermore, this is reinforced by being attached to more permanent parts of the cytoskeleton. So that now, that's sort of anchored. So that now our microtubules cannot just move up and down. So when we walk on one, this is going up, it's creating compression on this side. And it is stretching this side. And so this is happening all the way around. And so when we get that compression, it'll fold. Like that. Okay, so this is we're talking about hundreds of dynine motor proteins here. And it's really remarkable how they're all coordinated in order for the motion to cause the microtubule to bend. Further, the outside, plasma membrane. So unlike a bacterial flagellum, which is a naked protein structure outside of the plasma membrane, this is sheathed with plasma membrane. All these microtubules are inside of the plasma membrane, okay? So, quite a different structure. What makes them analogous is that in bacteria and also in eukaryotes, they have similar functions, but totally different structures, totally different evolutionary origins. Okay? And this is, just, we should have an animation here, yeah.
So the dynine on one microtubule is physically attached to the other?
That's right.
And so as it's walking...
It's physically attached to one microtubule doublet, but not to the adjacent one.
So it's walking along the adjacent one and it's
That's right.
attached to the other one?
That's right.
So it stretches.
And it's all that, all the way around,
Yeah.
you know. And so when one side is walking, the other side is relaxed. Okay, so we have these 9 doublets, and all those motor proteins are synchronized. Okay, it's a really amazing bit of control, and we don't quite understand how it works. Okay, all right. So there's other structures that are also microtubular. Something called centrosome centrioles. Okay, these are sort of related. First of all, let me talk about centrosomes. Okay, centrosome is an area. Neck, I already erased it. So I still have my cell. There we go. Okay, let's go over here. Get rid of the microtubules, or leave the microtubules there. You notice that I'm drawing all these microtubules coming from a point near the nucleus. That's the centrosome. So the centrosome is a microtubular organizing center, and eukaryotes have it, okay? So microtubular organizing center. And there's a lot of different types of proteins there. Now in animals, and some protists, within the centrosome is a pair of microtubular cylinders that have the same structure as a basal body. 9 peripheral triplets with no microtubules in the middle. Okay? That's the same structure as basal body at the base of flagellum. And they're short, and there's 2 of them at right angles to each other. Okay? Right angles. Okay? And they're within the centrosome. Okay? Now, most plants don't have centrioles. Now I said most because some of the older plants that evolved first, they do. Your textbook will say they don't, but they do. There are some. And likewise, most fungi don't have centrioles. They have centrosomes. They have centrosomes, but not centrioles within those centrosomes. Now, the exact function here, not sure. In animals, if you use a laser and destroy centrioles, usually cell division, not always, but cell division sometimes goes amiss, okay? So they seem to still have a major function when it comes to organizing the microtubules during cell division, all right? But still, plants and most fungi get along without them just fine, all right? So keep these 2 straight. Now, when I give definitions on quizzes, you really need to read each and every word I put there, okay? Just to give you an example, on the last quiz, one of the quizzes I mentioned, the first quiz, I mentioned a change in form and function of an individual over its lifetime. Some people put evolution. Individuals don't evolve, okay? Many generations evolve. Okay, so the 2 definitions of evolution, development sound very, very similar, but it's changes during the lifetime of an individual, that is development. So if you read it too fast and you miss that during the lifetime of an individual, it's going to lead you down the wrong road. Here's another example where that might happen. Centriole and centrosomes. They're not the same thing. Okay, related, very related, but not the same thing. Okay? All right, we characterize the structure, ultrastructure of microtubules in flagella, cilia, as being 9 plus 2, okay? 9 peripheral doublets and 2 separate in the middle. For basal bodies and centrioles, this is called 9 plus 0, but here the 9 stands for 9 peripheral triplets, okay? 3 microtubules fused together and none in the middle. So this is sort of the formula for these particular structures, all right? Okay. Okay, another structure in the cell walls. Animal cells don't have cell walls. Cell wall and plasma membrane, cell membrane, not the same thing. And so be real careful not to use the word cell wall when you're really talking about a phospholipid bilayer that surrounds the cell. So this is sort of a relatively non-dynamic structure, rigid outside of the plasma membrane, okay? And usually it's made out of structural polysaccharides in most of these things, plus other things as well. The plasma membrane is very dynamic. It is being changed constantly, constantly. Stuff is being brought from the Golgi and added to it. Stuff is being removed from the plasma membrane through endosomes, all right? And in plants, this here is the cell wall, fibers of cellulose. Okay, I mentioned that before. Okay, so this is a structural polysaccharide, and they form good fibers that, because of the hydrogen bonding that happens between adjacent beta glucose units, we have a real rigid structure that doesn't rotate, all right? And of course, this is another type of structural polysaccharide that I mentioned before, chitin. It's a polymer of N-acetylglucosamine, and it's beta N-acetylglucosamine. And so we have a similar story here. It is a good structural fiber because we also, besides the glycosidic linkage that holds the monomers together, we also have hydrogen bonding to make that a rather stiff fiber. And bacteria, they have something a little bit more complex called peptidoglycan. Whenever you see this word glycan, you need to think sugars, okay? That's carbohydrate. So yeah, there's structural carbohydrates here. We don't need to go into details what that structural carbohydrate happens to be, but they are cross-linked by short peptides, okay? Amino acids, maybe about 3 or 4 that they cross-link and make sort of a network for their cell walls. Animals do not have cell walls. Many protozoans, what we call protozoans, also do not have cell walls. The function? Protection, to a certain extent. And it gives the cells its shape, because it's relatively rigid. And especially in things that are living in fresh water and in bacteria, okay? It prevents excessive uptake of water. This isn't too much of a problem in marine water, but in fresh water, that is a problem, because no living organism can have an internal solute concentration that is below that of fresh water. And therefore, osmosis is a constant problem coming in. And if we have something that doesn't have a cell wall, then as the water moves in, it swells and will eventually burst. Now, we saw earlier that paramecium have what's called contractile vacuum. They collect the water and have to constantly be pushing it out. Okay. With plants, it's a different story. That cell wall prevents excessive uptake of water, and it's also true of bacteria. In fact, penicillin, how does penicillin work? Okay, this is an antibiotic that kills bacteria. It prevents the bacterium from making peptidoglycan cell wall. Okay, it interferes with that process. If they can't make that cell wall, then they're subject to being burst by water moving in, because a cell wall would push back, okay? It prevents excess water uptake. And so this is the function of cell walls. Okay? In addition to a cell wall, animal cells have still something outside of the plasma membrane called an extracellular matrix. And in some of the tissues in our body, the extracellular matrix can be quite extensive. Okay, for example, in connective tissue, have few cells, lots of extracellular matrix. And so this is a web, loose web of structural polysaccharides and proteins, and it's associated with the outer face of the plasma membrane. Those proteins and carbohydrates are made by the cells that live in that tissue, and they excrete it outside of the cell. All right? And what is function? To a lesser extent, also support, but not so, it's not strong enough to prevent osmotic problems, okay? Animals have to do it differently. A lot of this stuff is also for adhesion. Cells have to adhere, hold on to other cells or to the environment in which they are living so that they do not get carried away or moved around. So that's some of the function, and also animals regulation, cell communication. These things are involved. Some of the proteins here. We'll talk about this is a cartoon from your textbook, sort of showing that. Here's the plasma membrane. These are proteins, collagen fibers, proteoglycans. Okay, these feather-like things, those are branched carbohydrates, structural carbohydrates. And so you usually find them on the outside of the cell. Never on the inside, except for in the lumens of certain organelles, but not in the cytosol. Okay? Those are all the structures that I want to cover as far as typical cells. We'll add to these, of course, as we go on. That's one of the things about biology, is it keeps building, building, building. We'll add to it more and more. So any questions before I change pace here?
You said some exceptions in the beginning, is that what?
Yeah, so you can find structural carbohydrates sometimes on the lumen side of organelles, but not on the cytosol side. Okay. All right. Okay. So I'm going to switch gears here. Since we're talking about membranes, organelles that are membrane bound, we need to look more closely at membranes, biological membranes themselves, and what their function happens to be. So a little bit of history, I always like history. Okay, most of what we, early studies on biological membranes concentrated on studying the plasma membrane, because the plasma membrane was accessible, it's on the outside. A lot of these membranes on the inside were not accessible. They are now. We now can do a lot of stuff in understanding those. But nonetheless, what I'm going to mention here is some things, properties that were first of all discovered with the plasma membrane, and we know now that these attributes also apply to internal membranes as well. So, it's a very thin structure, real thin, 8 nanometers, okay? You really need to get a grasp on what a nanometer is. That's real small, okay? So they're very thin. And of course, plasma membrane is the boundary of the cell, okay? Completely surrounds the cell. It is involved in traffic in and out. So it controls what nutrients can come in, what waste can go out. And it is a selectively permeable structure. By saying selective permeability, we're referring to what some solutes can easily pass through it from one side to the other, whereas others are prevented from moving. So it's selective. Now the structure of these membranes, way back in the 1960s, they knew that it consisted of phospholipids and proteins, but they didn't know how those phospholipids and proteins were put together. There were several models that were suggested with protein on the outside and one layer of phospholipids on the inside, other models where it was just the other way around, et cetera, et cetera. They didn't quite know exactly how this was put together, but later they understood it, and it's been substantially supported ever since. So phospholipid bilayer, the phospholipid part of it, it will form spontaneously when you put it in the water. So we don't have to have enzymes constructed, all right? Because just simply due to the nature of the phospholipid, it has a polar end, hydrophilic, and then the two fatty acid tails, hydrophobic. They arrange themselves in the presence of water to be stable, the lowest energy conformation. And so that is a phospholipid bilayer. So those hydrophobic areas are forced internal, where they're out away from water. And they form spontaneously, but it's driven by water. If you put these in something in a non-polar solution, the phospholipid bilayer will fall apart. Okay, it won't hold together. Okay? Furthermore, there's proteins. In fact, the mass of most membranes, about 50% of the mass is phospholipid, 50% is proteins. And usually, it is the proteins that give that membrane its function. Not always. There's also some lipids that are functional as well, but broad generalization. Okay? All right, so this, once again, is a cartoon. showing phospholipid bilayer, protein, various types of proteins associated with the surface. Some of these proteins go all the way through, and so forth. And in this lecture, I'll talk about these proteins in addition to the phospholipids. But first of all, what is the actual structure historically? Is that back in the 1960s, they didn't quite understand exactly how the phospholipids and the proteins were arranged together. But in 1972, two scientists, Sainer and Nicholson, they came up with what is called the fluid mosaic model, which is accepted today and resoundingly has been experimentally supported. So instead of having a phospholipid layer and a protein layer, or several phospholipid layers and several protein layers, they're mixed together, making it a mosaic. Okay, so you have a phospholipid bilayer with the proteins in it and on both sides. It isn't a matter of a solid phospholipid layer here and a solid protein layer here. It's a mosaic. They're together. Furthermore, it's fluid. It's fluid. In fact, this here is a two-dimensional liquid. A viscous two-dimensional fluid. All right? So, now what stabilizes this, especially with proteins, they have to be happy in the membrane, and so there's hydrophobic interactions. So when we have these proteins that go all the way through the phospholipid bilayer, what type of amino acids do you expect here on those proteins? Hydrophobic, okay? And in fact, a lot of times, I'll get back to this a little bit later, a lot of these proteins, when they have a strand that goes through the phospholipid bilayer, they assume an alpha helix. And I'll explain why a little bit later, okay? What about out here? What type of arteries do you expect on the far inside, far outside? Hydrophilic. Yeah, okay. So those types of interactions, so there you'd have hydrophilic interactions, ionic, as well as hydrogen bonding on the outside, okay? Now, what type, I said that this was a liquid, okay, a viscous liquid, and so what type of liquids move, all right? They're not solid, not fixed in place in a crystal. They can move. And so what you get here is lateral drift within the monolayer, sideways movement, okay? You get that type of motion. Okay, for phospholipids, they tagged them experimentally, tagged them with a protein with fluorescent dye on it, and they could measure how fast they move under most conditions, and this is about 2 micrometers per second. So that's fast enough that within a couple of minutes, that phospholipid could have gone from one end of the cell to the other. Okay, so we're moving quite a bit. Proteins are slower, simply because proteins are much more massive. Okay, and therefore they move slower, but they do move. They're not fixed in space. But they can be. Okay, and I'll explain why. What is very rare is for flip-flop. Okay, that is changing monolayer sides. Okay, so for this phospholipid over here to switch from one monolayer to the other monolayer, we have that polar group, which consists of a phosphate with 4 oxygens, 2 of which are negatively charged, and another polar group attached to it, plus the glycerol. That is really a big impediment to forcing its way through a hydrophobic area. And therefore, flip-flop is rare. They can move laterally, but flip-flop to the other side is problematic. It happens on average for one particular phospholipid on average once a month. That's all.
So it's the entire phospholipid that's moved.
They can move within the monolayer. Yeah, on one side. And this can move in a monolayer on one side. Okay, but flip-flop is rare. About once a month, as I said. Okay, now, how originally did they figure out that this components of phospholipids moved? Well, the experiment was a clever experiment done in 1970 by Frey and Denon. What they did is they took a mouse cell and a human cell, and they labeled some of the proteins on the surface with fluorescent dyes, a red dye and a green dye. And they used something called a sonicator, which vibrates things quite a bit, and you can get these two forced together to fuse, all right? So they fuse these hybrid cells together. Now, we have two possibilities, okay? The membrane is fluid, or the membrane is static, stay still. Both of those hypotheses will make a prediction. If it's static, what do you expect to see 40 minutes later after you fuse these together with regard to red color, green color, if it's static?
It should be the same.
They would still, red would be on one side, green would be on the other side. It's a prediction, okay? If it's fluid, what do you expect? All mixed up. That's a prediction. Okay, this is what science does. And then they put together the experiment, looked at it 40 minutes later. What was observed was it was all the dyes were mixed, okay? So that demonstrated that things move, okay? Fluid model. And now we have other sophisticated ways, it's pretty well known. Okay, so what affects the fluidity? Because these membranes, if it's a liquid, it can be more fluid or less fluid, all right? Depending on the chemical nature. So if we just only have phospholipids, just talking about phospholipids, one thing that affects the fluidity is the nature of the fatty acid tails, whether they're saturated or unsaturated, okay? How would these, if we're talking about phospholipids, where the fatty acid tails are the same length, okay, all of the same length. But here we have one membrane that has saturated fatty acids, another one that has a fair amount of cis unsaturated fatty acid. Which one of those two would you predict to be more fluid? Which one?
Unsaturated.
One with the cis unsaturated fatty acids, okay, because of close packing, okay, that holds them together, makes them viscous. All right, that's one factor. Okay, they're more fluid, they have the kinks, less fluid if without them. All right, so these are going to hook together too much. We have a stronger hydrophobic interaction here than here. Okay, that space. Okay, close packing. That's one factor. Second factor is the length of the fatty acid. Yeah.
Is it possible that with temperature changes that you can go from saturated to unsaturated?
That's exactly what they do. Yeah, so I'll get to that. Because these membranes, remember homeostasis, if the temperature changed, the membrane has to change. If it's going to stay at the same viscosity, okay?
Would it make a difference if it was a trans-unsaturated fat?
Trans-unsaturated fat would be just like saturated fat, okay? Because those are straight chain. Okay, yeah, those are straight chain. Okay, length of the fatty acids, okay? Why would that have any effect? And in what direction? Long versus short.
Shorter makes it more fluid because there's less mass to move around.
Say what?
Shorter makes it more fluid because there's less mass to move around.
Okay, what did you gonna say?
The surface, there's no surface.
The longer ones have more hydrophobic surface in contact, stronger stickiness, okay? If they're short, then you don't have as much of a hydrophilic. Now, here we're assuming that they're unsaturated, okay, unsaturated. Long chains have more surface in contact, a stronger hydrophobic attraction. Short surfaces, not as much, okay? Because the strength of the hydrophobic interaction is proportional to the surface area in contact. And so, yes, that's a second factor, okay? So a third factor is the presence of sterols, okay? Sterols, so these, as I've already went over, they're lipids, that happen to have four hydrocarbon rings, right? So like with cholesterol, it is bulky, it has a hydroxyl group up here, okay, this is for animal cells. Animal cells have, in their membranes, they have a lot of cholesterol, all right? And so this is bulky, and so we have cholesterol on both sides. Now, plants and fungi, they also have sterols, but not cholesterol. They have different types of sterols. And since we animals have a problem with cholesterol, so it's testosterone in plants, it's still a four-ringed sterol, and fungi have ergosterol. All right, so this here, what role does cholesterol have? It actually influences the fluidity at phase transitions. Phase transitions. If you lower the temperature, you're lowering the kinetic energy of movement of the phospholipids. And if you lower it enough, then those phospholipids stick together, okay? It becomes a solid. That would be fatal, okay? If you increase the temperature high, you're moving, remember this is a two-dimensional liquid, it's a viscous liquid, but if you take the temperature up, you have so much kinetic energy that things can fall out of you melt the membrane, okay? It goes into sort of analogous to a gas. So this here works, cholesterol works at both transitions. And so having sterols at low temperatures increases the viscosity. Having sterols at high temperatures lowers the viscosity. Okay, so it has a dual nature at phase transitions. Now, why is that working? Okay, this ring, these four rings, they are planar. Okay, they're in one plane, and therefore they're holding and pushing apart these phospholipids. Yes, this is hydrophobic, but because of the shape there, we don't have too much of a hydrophobic interaction right here or here. It's only where the cholesterol is touching, okay? And therefore, this would tend to make it more fluid, all right? Up here, that hydroxyl group that's at the surface, it can hold hydrophilic heads together. And so that's what comes into play when temperature gets too high, all right? So plants, they have to alter their plasma membranes with the season, and even during the day. When the temperature is cold, because they have to keep their viscosity of the membranes within homeostatic balance. Okay, if it gets too hot, it gets too fluid, it makes changes. It removes material from the plasma membrane and puts other things in there. So what changes, if we're at too high of a temperature, what changes can the plant cell make to alter its fluidity? Three changes. Tell me three changes that it can make.
It could add more cytosterol into its membrane.
What?
It could add more cytosterol into its
That's always a good answer. You can add sterols, okay? That would help up to an extent. What other alterations would slow it down?
Increasing the amount of unsaturated fatty acids.
Increasing the amount of unsaturated or saturated fatty acids. So exchanging, removing phospholipids that have unsaturated fatty acids and replacing them with phospholipids that are saturated fatty acids. What other? One more.
add longer.
Yeah, longer fatty acids. Okay, longer fatty acids. All these changes would serve to decrease the viscosity. Okay, and yes, membrane structure is constantly being altered. Okay, these things are not static. Okay, to maintain homeostasis, those are the changes that have to go. All right, so I already said this, less fluid at high temperatures. All right, now, so plants have to alter their composition, maintain that that's what I just went over. So the membranes are constantly being changed. And if it's a cell that is not growing, if it's a cell that's maintaining the same size, just as much, remember we have all this traffic that's coming from the Golgi and fusing to the plasma membrane, but if the cell is not growing, the same amount of membrane has to be removed through endosomes, okay, coming in from the plasma membrane in order to stay in balance. Yes?
So wait, at higher temperatures, you wanna make the membrane more viscous, and at lower temperatures, you wanna make it more fluid.
Say that again.
So at higher temperatures, you wanna make the plasma membrane more viscous,
That's right.
and at lower temperatures, you wanna make it more fluid.
That's right.
Okay.
Yeah, yeah. Okay, so this is always changing. Now, with animals that maintain, like ourselves, homeotherms, we try to maintain a constant body temperature, all right? But still, the membranes are changing, not as much as with plants. Okay, so some of these, what about the proteins? Okay, that are associated with membranes. I'm gonna put them in 3 categories, and actually 2 categories. Some of these are integral proteins, and they have a strong attachment to the membrane. They can't be removed very easily for study, all right? And there's 2 groups of these, so they have hydrophobic, so these integral proteins either have, yes?
Sorry, could you go back, because earlier you said lower temperatures increase viscosity, and then when she asked, and then you said that it was.
Okay, maybe I misunderstood her. So what was it?
So, you had said more fluid at lower temperatures because it increases the viscosity. And then she said at lower temperatures it increases.
Okay, so at low
fluidity.
temperatures, we want to increase the fluidity. At low temperatures, you want to increase the fluidity.
So increase viscosity.
Yeah. Okay, at high temperatures, it's already too fluid, we want to slow it down,
Yeah,
okay, make it more viscous,
okay.
okay? Yeah, okay? Okay. So membrane proteins. When I'm talking about integral proteins, they are strongly attached. Either they are embedded in the hydrophobic region, or they are covalently attached to something that is solidly embedded in the membrane, okay? So I'm gonna give 2 different categories here. Your textbook doesn't do this, okay? I'm giving 2 different categories. Transmembrane integral proteins, they go all the way through, okay? In fact, they work on both sides of the membrane. They operate on both sides of the membrane. And as I said, when we have, so these bars here are, or these cylinders, those are alpha helices. This is very, very common for when a strand of the polypeptide is snaked through the membrane, it assumes an alpha helix. Okay, we already understood that most of the R groups there are hydrophobic, right? Okay, but what about the backbone? What do we have along the backbone? NCC, what's on the end? Is that polar? How about the C end? Has double bonded oxygen? Those don't like being, those have partial dipoles, positive charge and negative charge slightly. They're not gonna like being in the membrane, but if they associate with each other with hydrogen bonds, neutralize it. You get it? If they have hydrogen bonds, we have all those things associated with hydrogen bonds, they neutralize each other. And they're happy, okay? So this is the reason why we have alpha helix when it goes through here. Now, this can be more complex, because some of these proteins, they may, well, I probably put it up here, we have maybe it's on another slide, okay? Yeah, single spanning or multiple spanning. What is a common motif is 7 spanning alpha helices. That's real common. So we have here, here's our phospholipid bilayer, and let's say N-terminus down here, have alpha helix there, unordered, another alpha helix here, and it goes through 7 times, okay? All right, I'm not gonna draw it. Now, if we happen to look at it from above, okay, so here's one alpha helix, another, 1, 2, 3, 4, 5, 6, 7. Some of these things can have a water tunnel through it. So along here, these are groups that are in contact with, so I'm doing a cross section right through here, okay, in the membrane. These R groups have to be what? Polar, hydrophilic, hydrophobic? Hydrophobic. But what about the ones that are over here? Those have to be hydrophilic. Okay, so as this is spiring, you know, the picture can get very complicated, even though there is an alpha helix. Okay, that sometimes happens. All right, so single spanning, multiple spanning. All right, we have both. And so this is still integral proteins. So I'm going to change a little bit from your book. I'm going to call this unilateral. Think that your book may use the word unilateral or peripheral and join these two together, but I'm separating them. Okay, this here is an integral protein, unilateral, it's on one side. It's one side of the membrane, and it is covalently bonded to a phospholipid or a glycolipid or something. It has a strong connection to the membrane. It is not necessarily in the membrane, okay? It may be on the outside of the membrane, but it is anchored there, okay? Can't be washed away and studied very easily, all right? So this here is still, but it works on one side, all right? Second group, so both of those transmembranes and unilateral membranes, those are integral, meaning strong attachment. This is where I deviate from your book. Peripheral proteins are also on one side, but detachment is weak. Okay, maybe a weak hydrogen bond or ionic bond, something along that line that can be easily be broken. Okay, so these proteins can be washed off. If we wash the membrane with a salt solution of the right concentration, we can remove the proteins so that we can study them. Whereas with integral proteins, that's not the case. Okay, so it's a matter of how strong the attachment happens to be. Okay, so this is from a different textbook showing some of these. What would you call that? Purple. So I'm going to start on the left, work my way through. Okay, that's a transmembrane integral, obviously. Single pass. Okay, over here, multiple pass, also transmembrane. Okay, what would you call this? This here is a sugar. That's a phospholipid. That's a protein. Unilateral, peripheral. Unilateral, okay, because it is covalently bonded to that sugar. The sugar is, or that oligosaccharide is covalently bonded to a phospholipid. So that's a strong connection. What about this one? Peripheral or unilateral? It's unilateral because that here is a lipid anchor. It's probably fatty acid. Okay, probably a fatty acid. It's not a phospholipid, it's fatty acid, but we have a covalent link there. So there is a lipid anchor that keeps it there. What about, okay, this is of course a single pass transmembrane protein, but what about this one? Okay, that's peripheral. Okay, there's no covalent bond, and so forth. That's peripheral, purple one at the top. Okay, and so forth. This here, peripheral or unilateral? Yeah, part of it. Hydrophobic areas here that are embedded strong enough, it's embedded on one side. It has hydrophobic amino acids up here, the blue one, and therefore it has a strong attachment. Okay, so these are some of the general proteins, but what are some of the functions? Now, as we go through here, we're going to touch on all of these, so you don't have to memorize them now. Unless you want to, you'll be ahead of the game, okay, because we will talk about all these. Some of these allow passage for things that normally cannot go through the membrane, and therefore they're, do that. Most receptors for communication happen to be transmembrane proteins, okay? Transmembrane proteins convey chemical signals. Some of these are enzymes that catalyze reactions at the surface of the membrane. Some of these proteins occupy, they hold cells together, okay? Later in even in this lecture, and also very strongly in Biology 101B, I'll be talking about a type of tissue, epithelial. In animals, it's an important tissue, but the cells are very closely packed together. Not a lot of space between them. And furthermore, they have proteins that go from one plasma membrane to the next to hold them together, so that this sheet of cells makes a sort of a barrier, and you're now dividing two different compartments from each other. And I'll have more to say about that later. So adhesion proteins, tight junctions in animals or vertebrates, they're called something else in non-vertebrates. Some of these proteins are also involved in communication, okay, cell communication. Energy transformation, we'll see a lot of examples of that in photosynthesis, cellular respiration when we get there. Okay, now these membranes, okay, so this is another story, all right? These membranes are like politicians, they're two-faced, okay? So these are bifacial. They have the two monolayers are different, all right? They don't have to be identical. They can have different lipids. Now, we haven't gone into naming all of these different phospholipids, but indeed, we have a lot of phospholipids that they have special names. And each side can differ in their lipid composition. I'll give you just one example. This is sort of an important example as far as your immune system is concerned, phosphatidylserine. So this is a phospholipid in which the polar group is an amino acid serine. Okay, that's an amino acid. You only find that on the cytosolic side of the plasma membrane, never on the external plasma membrane, unless something's wrong with the cell. And this is how some of your immune system cells can tell whether or not this particular cell has been infected by a virus. Because when the virus gets in there, it will invert some of the components of membranes. And if your immune system cells find phosphatidylserine on the outside, they will kill the cell, okay? So that's important. Now, I said things happen to once a month, flip-flop. So does this sometimes end up on the external monolayer? Yes, but there are proteins in the membrane that can detect that. They're called flip-flopases, believe it or not, and they will flip it back over, okay? A more scientific word for those. Phosphatidyltransferases, but you can call them flip-flopases too, okay? All right. It works. You actually find it in the literature too. They call them that, all right? Okay, so, all right, furthermore, the different proteins. Each side of the monolayer can differ with regard to the proteins that they're made out of. Furthermore, these transmembrane proteins usually have directionality, and furthermore, they have to function different from each other. I already mentioned receptors, okay, like in hormone systems. How does a hormone is a chemical signal, and for a cell to respond to that chemical signal, it has to have the right receptor. If it don't have a receptor for that chemical, that hormone, then the cell can't even detect it. All these receptors are transmembrane, okay? And furthermore, let me draw a picture. They have to have on the outside a hormone binding site, okay? So on the outside, it has to have binding domain. When I say domains, these are subsections of a protein or polypeptide that has a folding pattern that it functions as one particular unit, okay? It has a special function. And so this has three domains. We have a binding domain, a transmembrane domain, and then maybe a catalytic domain on the inside. So when the hormone binds, that leads to a change of conformational shape. This is going to be a theme we have all through biology. When you bind something to a protein, you usually will end up changing its conformation. And some of these proteins have several different native conformations. That's how they work, okay? And so this will change its conformation, and this catalytic unit will change, and there's other proteins in here that it affects their behavior. It causes them to change shape, and it changes everything that's happening inside of the cell, okay? So this here, if we put this protein in there the other way around, it's not gonna work. If we put the binding site inside, it'll never see the hormone, okay? So the two faces with the membrane with regard to their transmembrane proteins, they have different functions there, okay? Furthermore, carbohydrates that happen to be there will be different. With the plasma membrane, you find branching oligosaccharides only on the exterior face, never on the inside cell face. Okay, some of these are cell markers. Hey, this is me, okay? Don't kill me, immune system, all right? And others are, with communication. So some of these proteins can be glycoprotein, a protein, sugar-coated, okay? A glycolipid, that being, okay, that's not a protein, we're talking about carbohydrates, sorry about that. All right, so we can have a sugar unit, oligosaccharide attached to a protein, or a sugar unit attached to a lipid, and those can differ from one side of the membrane to the next. They can even have different fluidity. The two monolayers work fluidly separate from each other, okay? And so all these distinctions can happen. That's what we mean by the membrane being bifacial. So composition, they can differ from organelle to organelle. They usually do. The organelles have different membrane proteins, because usually it's the proteins that give them their function, okay? Furthermore, from cell type to cell type, okay? If we're talking about the same membrane, so a plasma membrane of a red blood cell will be compositionally different in certain respects to the cell membrane of a muscle cell, okay? And from species to species, the plasma membrane of a red blood cell in a chimpanzee may not be exactly the same as in a human. Yes?
On the last slide, or the different fluidity, does that have to do with the mosaic membrane?
Well, yeah, this is still, all of this is part of the fluid mosaic model. Yeah, that's right. Because the membranes are fluid, but the two monolayers don't have to have the same fluidity. Okay, they can differ. Okay, all right. So, things are made in the rough ER, smooth ER, shipped by transport vesicles to the Golgi, from the Golgi to other membranes. Some of these transport or transmembrane proteins, they have a very definite directionality. And so when they are first inserted into a membrane at the rough ER, they already have to be oriented in the same right direction to work where they end up at. Okay, their final destination. So it has to be correct and it has to start from the very beginning. And so all this movement of membranes from one organelle to the next follows what is called the principle of topological equivalence of membranes. Topology does not mean chemical composition. Topology means the surface, and we're talking about two different surfaces. There's a surface that's facing the cytosol, or there's a surface that's facing the lumen of the internal organelles. With regard to the plasma membrane, we have a cytosolic surface, and we have an external surface. And so that's what we're talking about, not necessarily chemical composition. So this here states that the side facing the cytosol, as we move from one organelle to the next, transport vesicle, Golgi, transport vesicle, plasma membrane, that the monolayer that started out facing the cytosol will end up facing the cytosol at its final destination. The side that's facing the lumen of an organelle will face the lumen all the way through the transport system, and when it fuses to the plasma membrane, it will be away from the cytosol. So the exterior surface, topologically, of the plasma membrane, the exterior surface of the plasma membrane is topologically equivalent to the lumen side of all the organelles. Okay, all right. All right. I know you're scared, but let me explain. Okay, so let's go with a receptor. Okay, receptor. The binding, hormone binding site, well, when we go here, it has to be on the outside. The catalytic site has to be on the inside. This is the side facing the cytosol. This is the side facing away from the cytosol. And so it has to be so oriented that way when it is inserted in the rough ER. So here we have the rough ER. Let's say, although this is meant to be that green thing there, it's meant to be an oligosaccharide. Let's say that that is the hormone binding site. So rough ER, this protein was made by bound ribosome. It was inserted into the membrane. But what we have here is that the binding site is facing the lumen. The catalytic site is facing the cytosol. It's on the cytosol side. When this buds off as a transport vesicle, it's the same orientation. Okay? When it fuses to the Golgi, same orientation. Our binding site is on the cytosol side, and the catalytic site is on on the lumen side, and the catalytic site is on the cytosol side. When it's in that transport vesicle, same orientation. When it binds to the plasma membrane, notice how this binds. First of all, we'll make contact, and then the two bilayers will fit together so that the inside is flipped outward. So what was once the lumen side of the transport vesicle is now facing the outside of the plasma membrane. And we have our binding site outside and catalytic site inside. Okay? I'll do one more. Antibodies. Antibodies. These are defensive proteins. Where do they function in the body? Outside the cell. Okay? So they're not in a membrane at all. They're not in a membrane at all. They have to be secreted. So they're proteins, rough ER, they're inserted through and they will be in the lumen. They will be in the lumen, okay? And then when that's a transport vesicle, they will be in the lumen. When it binds to the Golgi, it'll be in the lumen. Transport vesicle, it'll be in the lumen. When that binds to the plasma membrane, it's dumped outside. Okay? So this is topological equivalence. Those membranes don't flip over. We get an inversion at the plasma membrane when they fuse, but the side of the membrane that was facing the cytosol will always face the cytosol. The side that's facing away from the cytosol will always face away from the cytosol. And this is how the movement happens to work. this, I'll stop right here, and I'll continue this on Thursday. Okay, any questions? Plot thickens, right?